With the work showing that we've made progress, we've improved the median survival for malignant gliomas significantly over the last decade, we now have a very extensive pipeline of new treatments and new approaches and new delivery methods that are being evaluated in clinical trials. And with that, and with the fact that we have seen progress, although it's not enough, that we have seen progress with this approach, I'm very optimistic that with some of the newer approaches that we'll see even more success. Our work with using interstitial chemotherapy, with using polymers that release chemotherapy, has been focused primarily on malignant gliomas in adults. That's the patient population that there's really no effective therapy. The median survival was 9 months, no matter what was done when we started. And now the median survival with local delivery plus systemic chemotherapy, Timidar, plus radiation therapy, and plus the advances in surgery and in intensive care is now more than 20 months. And whereas before it was rare to see a long-term survivor, now it's common, and 20–25% are living several years after diagnosis. So we're seeing a different outcome, which is encouraging because it really is a change in the outcome for this terrible disease. But it's still, of course, very unsatisfying in that it's still a terrible disease and it's a terrible outcome. We could do much better. Nonetheless, the fact that we've made progress means that we should focus on children now and bring these advances to children, which is a harder population to treat, and we should use much better both drug delivery systems that are being developed and much better therapies. Well, when we first started, the concern was, could you put very high concentrations of drugs directly in the brain? There was concern that it might be toxic. These are very strong drugs that have toxicities, so the question was it safe to do this? Also, was there a mechanism that you could even do this with? So Bob Langer's professor at MIT developed a biodegradable polymer that, in fact, he showed was safe and could release in a controlled manner over several weeks instead of over minutes, the way we currently give drugs intravenously. And then we showed that it was safe in the brains of first small rodents, and then higher animals, and then in 1987 we began studying it in people in a dose escalation study and showed that in fact it was safe in people. We then, the criticism was, well, it works in animals and it's safe, but does it really, is it really effective in people? Will it be effective? Anybody can cure cancer in mice, but would it really make a difference to people? So what we elected to do, and was very unusual at that time, is we used the same rigor that we used in the laboratory of doing carefully controlled studies. We did it with people, and we did a randomized, prospective, placebo-controlled study. We put polymers with no drug and polymers with drug and otherwise best available therapy. And first in 222 patients who had failed all other therapies, end-stage adult malignant gliomas. And we showed with that in fact there was a 50% improvement in survival with that one variable of having the polymer with the drug in it. And on the basis of that, we expanded to using it as initial therapy with studies here in Europe, as well as other studies in the United States. We modeled out that there was a benefit, that it was safe and effective, and we did all the pharmacokinetics in animals, and first in vitro and then in animals, and we really were very confident that at least experimentally we could show a strong benefit. Then the question became, could we design clinical studies that truly paralleled what we had learned in the laboratory, and to see whether the benefit translated to people. And we are very pleased with that in fact the survival curves are almost superimposable, that you see the same percentage of improvement in survival and the improvement in long-term survival in the experimental models as we subsequently saw with people in phase 3 studies. The brain has a way of protecting itself. It's called the blood-brain barrier, and it's put there by nature so that things that could harm the brain, toxic substances that might be in the environment, don't get into the brain. It's a very protected site. So a lot of the therapies that we do to harm the cancer could be harmful to the brain. And so the brain has this wonderful mechanism for blocking our therapeutic agents from getting into the brain. So drug delivery systems are very important because they allow us to bypass the natural defense of the brain against drugs that or other therapies, immunotherapies, biological therapies that we need to get into the brain so they can reach their target site. We now have more effective agents. We have biological agents that have never been tried before. We have tremendous knowledge of the molecular genetics of childhood cancers and adult cancers that allow us to target specific overexpressed genes that we might be able to then benefit those patients with those very targeted therapies. And we do have multiple methods of delivering therapies directly into the brain. We have polymers. We have all sorts of additional polymers other than the FDA-approved gliadel polymer. We have microchips where we can place multiple drugs simultaneously into the brain and then release them in a controlled fashion externally. That's at a very early stage of development, but we're working on that, and certainly we've done that successfully in the laboratory. And there's convection-enhanced delivery and methods of infusing under pressure drugs to get them into the brain, and also methods of disrupting the blood-brain barrier so that you open up that barrier, deliver the drug that's necessary, and then close the barrier again. So scientifically, we've advanced significantly, and now it's a question of developing the clinical trials to make that beneficial for patients. Well, these workshops are important because they cross-fertilize across age groups. The tumors that children get are special. They are similar biologically. The brains pose all sorts of different problems. The developing brain, in some ways, is more resilient than an adult brain. In some ways, it's much more exquisitely sensitive to damage because the development is so critical. So every age of the child's brain tumor poses a different set of advantages and disadvantages that need to be taken into account. But I think the major thing is that we sit down and we talk and hear what's been successful and what's not been successful, and we cross disciplines and learn how to do things better. It's a huge advantage. We'll say that it's not only the pivotal papers and studies that showed the benefit of using these chemotherapy wafers, but now there have been meta-analysis involving over 6,000 patients that have been in trials with chemotherapy wafers. And if you do a meta-analysis, the overall survival is significantly improved over standard therapies. And I think that leads to a very strong argument that this should be added routinely to the armamentarium in combination with systemic chemotherapy to help adult patients with malignant gliomas. And I think that it behooves us to extrapolate that to children and safely be sure that we can do similar approaches for these very devastating diseases.